Dear students, very good morning. Today we are going to discuss about one of the novel drug delivery systems such as liposomes. The liposomes, that means that is composed of vesicles of bilayer of phospholipids. It has a lot of applications in the pharmaceutical field. Let's see one by one. First we will see the definition of liposomes. Liposomes are microscopic spheres with an aqueous core surrounded by one or more outer cells. It's consisting of the lipids arranged in the bilayer configurations. So the liposomes is a spherical vesicles with a membrane composed of phospholipids bilayer used to deliver the drugs. There are the several drugs that has to be delivered in the form of liposomes to avoid the side effect or genetic materials into the cell. Okay, we can incorporate with the drug into the inner core of the liposomes. The liposomes can be composed of naturally derived phospholipids with the mixed lipid chain like egg, phosphorylethanolamine, or the pure components like DOPE, like diolyl phosphoryl ethanolamine. So likewise we are incorporating with the cholesterol components. Then the liposomes, it's generally it is a tiny bubble the made up of same material as well as the membrane. The liposomes can be filled with the drugs. Okay, as I told you earlier, it can be filled with the many drugs. Which drugs we can easy to dispose it to the particular site of the body, so that we can load into the drug in the liposomal formulations, especially for the cancer drugs and other disease also we can administer in the form of liposomes. So liposomes were discovered by the British hematologist like Dr. Alag D. Bangham in the year of 1961 at the Bangham Institute of Cambridge. So they were discovered the Bangham and R.W. Hahn were testing that institute the newborn, so new electron microscope by adding the negative stain to dry phospholipids. The resemblance to the plasmalemma was obvious and a microscopic picture was observed. As the first revealed the evidence of the cell membrane the being bilayer structure. Okay. So see the structure of the liposomal drug delivery system. So the drug to be in the part of the inner core. It is surrounded by the aqueous layer, and aqueous layer is surrounded by the bilayer of phospholipids. That hydrophilic head. It is inner in the aqueous solution and hydrophobic tail in the outer layer. Okay. This is the various diagrams. There are the different classifications of lamellar, unilamellar vesicles, large unilamellar vesicles, small unilamellar vesicles, and multilamellar vesicles. They have a different size. There are the different types of liposomes. When we depending upon the structure, there are two types of liposome. One unilamellar liposomes, another one is multilamellar liposomes. So unilamellar liposomes, the vesicles as a single phospholipid bilayer spheres enclosing that aqueous solutions. The multilamellar liposomes, that means the multilamellar vesicles have an onion like structure. Okay, so it's an onion like structure. The typically the several unilamellar vesicles will form a one inside the other, diminishing the size and creating the multilamellar structure of the concentric phospholipid spheres separated by the layers of water. See the different structure of liposomal types. Then usually the composed of cholesterol and phospholipids, the structure, the composition proportion are being practically as the same in the host cell membranes. It may be vary with the method of preparation. The phospholipids possess the hydrophobic tile structure and the hydrophilic head component and organizing the following when dissolved in the water. Okay, the hydrophobic tail mutually attract while the hydrophilic heads contact with the aqueous solutions. So it have a lot of advantage as a drug delivery systems. It has a lot of advantage. It provides a selective passive targeting to the tumor tissue. As like when we treating that the tumor disease, we can incorporate that drug in the form of liposomal, like liposomal doxorubicin. They increase the efficacy and therapeutic index, and also it is increased the stability via encapsulation, because that the inner core of the drug, it is encapsulated with the bilayer of phospholipids. The reduction in the toxicity of the encapsulated agents and the side avoidance effect, the improved pharmacokinetic effect like reduce the elimination, increase the circulation times also. Okay, the flexibility to couple with the site specific ligands to achieve the active targeting. So these are all the advantage of liposomes. So it have the many advantage, so when compared to some of the drug delivery systems. So even though it has a lot of advantage, but it has a few drawback also. The leakage of encapsulated drug during the storage. When you storing the drug, when you storing the liposomal formulation, there may be chance to leakage. So that is a major drawback of the liposomes. Another one is the short half-life and batch to batch variations, the difficulty in the large scale manufacturing and sterilization. So it is easy to way we can prepare the small scale, but when you are going to manufacture in the bulk unit, it is difficult. Okay, the production cost is high compared to some other dosage forms. Once administered, the liposomes cannot be removed. That also another big drawback. Sometimes the phospholipids undergo the hydrolysis and oxidation reactions. These are all the drawback of liposomal preparations. Let's see, there are the four different mechanisms of the liposomal formulation. The endocytosis by phagocytic cells of the reticuloendothelial system, such as macrophages and the neutrophils. That is endocytosis by the macrophagic cells. Second one is absorption to the cell surface either by non-specific weak hydrophobic or electrostatic forces, or by the specific interaction with the cell surface components. The third one is the fusion with the plasma cell membrane by the insertion of the lipid bilayer of the liposomes into the plasma membrane, with the simultaneous release of the liposomal content into the cytoplasm. Okay, the another mechanism, the transfer of the liposomes lipid into the cellular or subcellular membrane, or vice versa, without any association with the liposomal content. Okay, so these are all the four different mechanisms of the liposomes. It is often difficult to determine at what mechanism it is operative, and sometimes it may be involved in the two different mechanisms at the same time. Okay, so it is very difficult to determine at which mechanisms the drug is going to release out of these four mechanisms. Okay, so these are the structure based liposomal preparations. Multilamellar vesicles, then oligolamellar vesicles, the unilamellar vesicles, then multilamellar vesicles. Okay, so they may be have the multilamellar vesicles greater than the 0.5 micrometer, then oligolamellar vesicles, it is 0.5 to 1 micrometer, then unilamellar vesicles, there are all the different size range. So again it is further classified into medium unilamellar vesicles, small unilamellar vesicles, then giant unilamellar vesicles is greater than 1 micrometer. Okay, then large unilamellar vesicle, it is greater than 100 nanometer. So based on that, based on the structure size, the liposomes are categorized. There are some correct size of the liposome preparation depending upon the following parameters. So when you going to prepare the liposomes, it is a challenging one. The physiochemical characteristic of the material to be enwrapped and those of the liposomal ingredients. Second one is nature of the medium in which the lipids vesicles are dispersed. Okay, என்ன medium select பண்றாங்கிறது depends இருக்கு. Okay, the effective concentration of the enwrapped substance and its potential toxicity, the additional process involved during the applications and the delivery of the vesicles. Okay, the additional process involved during the applications and the delivery of the vesicles. The optimum size, polydispersity and the shelf life of the vesicles for intended applications, the batch to batch reproducibility and the possibility of large scale production of safe and efficient of the liposomal preparations. Okay, so before going to do any method of preparation of liposomes, we must know the physiochemical characteristic of the material to be enwrapped under those liposomal ingredients. Second one is nature of the medium, என்ன medium நீங்க select பண்றீங்க, in which the lipids vesicles are dispersed. Okay, then what about the concentration, effective concentration of the enwrapped substance and its potential toxicity. Okay, let's see, these are the various methods of liposomal preparations. It was categorized into two way, passive loading techniques and active loading technique. Mechanical dispersion method இருக்குதுல, solvent dispersion method இருக்கு, then detergence methods. So mechanical dispersion by using the mechanical instruments, by liquid film hydration handshaking method, non handshaking method, then micro emulsifications, then sonications, uh, French pressure cell, then cell membrane extrusion technique, the dried reconstituted vesicles, and frigida liposomal preparations. Okay, likewise when we the solvent dispersion, here we are using that many solvent, like organic solvents, ethanol injection method, ether injection method, the double emulsion vesicles, the reverse phase evaporations, and the stable plurimellar vesicles. Okay, this is another method, we prepare the liposomes by solvent, by using the solvent. Another one, the detergents removal methods. The detergents removed from the mixed by dialysis method and column chromatography, or by dilution, or by reconstituted virus enveloped vesicles. Okay, then general methods of preparations. So generally the liposomes are prepared very simplest way. The liposomes are manufactured in majority of the way using various procedure in which the water soluble drugs are enwrapped by using a aqueous solution of this material as a hydrating fluid, or by adding the drug, or adding the drug solutions at the same stage during the manufacturing of the liposomes. Okay, this is the initial process. Then the lipid soluble materials are solubilized in the organic solution of the constitutive lipids. Then it is evaporated to dry drug containing the lipid thin film is followed by the hydration. This is the basic method for preparing the all type of liposomal preparations. These methods involves the loading of enwrapped agents before or during the manufacturing procedure. This is called a passive loading technique in சொல்றோம். And let's see the diagrams. It shows that however the certain types of compounds with the ionizable groups, and those which displays both the lipid and water solubility, it can be introduced into the liposomes after the formation of intact vesicles, that to be loading. Okay, general method, when we incorporating the cholesterol, the lecithin, and charge that to be dissolved in the organic solvent, then the organic solvent, the solution in the organic solvent, then allow to drying, it will get that a thin film is formed. Okay, then thin film, after formation of thin film, it is disposed in the hydrations, again the liposomes in the suspension. So finally we can purify the liposomes by the any one of the suitable technique. Okay, let's see the continuation of the topic in the next class. Thank you.